Welcome to Chapter 12 of Anatomy and Physiology, where we're going to do nervous tissue. And even though we're really talking about nervous tissue, it's really nervous system. So like usual, before you even get started, you know some things about this already, because you know some things about nervous system way back from Chapter 1. You at least know the organs here. It's only 3 of them. There's the brain, the spinal cord, and on this cartoon, all the creepy yellow lines you see on the guy are nerves. Those are the 3 organs: brain, spinal cord, and nerves. And because there are multiple organs here, we're going to have to break them up and classify them like usual, like we do in anatomy. So when we look at the nervous system, one way to break it up is structurally. We break it up into a central nervous system and a peripheral nervous system. And your central nervous system, it kind of gives you a hint of the name. Think of what organs are really in the center or the midline of the body. It's your brain and your spinal cord. They're representing the central nervous system. While all your nerves kind of go off to the periphery, so your nerves are what we consider the peripheral nervous system. And so when we look at these 2 systems, again, it's just a breakdown structurally. Your brain and spinal cord are central nervous system, and all the nerves in your body are peripheral nervous system. And it's the nerves that are doing a lot of work. Your nerves are helping to send and receive signals from outside of the body and send them to the brain and spinal cord in the central nervous system. But when you talk about sending and receiving information, it's going to be a lot. Remember, this system can also control other systems. So these nerves are sending and receiving signals from lots of different places. So when we look at the peripheral nervous system, we're going to have to break it down. And we're going to break it down functionally, based off what information those nerves are sending and receiving signals from. For example, one thing your nervous system can help you to do is to move. You could talk to muscles. Okay? For you to move, it begins with your nervous system talking to muscles. But you got to remember, you have different types of muscle tissue: skeletal, cardiac, and smooth. So when it comes to talking to each of these different types of muscle tissue, well, it's going to be the responsibility of different divisions of your peripheral nervous system. For example, if you want to talk to skeletal muscle, which is the muscle you use to move around, that's actually the job of what we call the somatic nervous system. But on the other hand, if you needed to talk to cardiac or smooth muscle tissue, that's actually the job of the autonomic nervous system. Remember, you do not control smooth and cardiac muscle tissue, but just because you don't consciously control it doesn't mean nothing could talk to it. It's just a part of the nervous system that you actually don't control either, that we call the autonomic nervous system. What else? And besides talking to muscle, you could even help to sense your environment. Remember, sensation is a function of nervous system. And when it comes to sensing things, again, you could sense them in different places. For example, if you're sensing general sensations like pain, temperature, light, touch, well, that's again the responsibility of your somatic nervous system. But on the other hand, if you were having more internal body sensations, let's say you wanted to feel the pain of a tummy ache, well, that's an internal body pain that you're going to have to sense. You're going to be able to sense internal sensations with what we call the enteric nervous system. That's helping you to sense a lot of internal body structures, especially in the digestive system. And also with the digestive system, not only that, you could also talk to the smooth muscle in your intestines. So when we talk about moving things around internally, especially for the digestive system or feeling it, it's all enteric nervous system. Think of that as the brain for your gut, is the enteric nervous system. So we'll talk to muscle and help with sensations there. But if you need to talk to skeletal muscle, remember that's somatic nervous system. If you need to feel things on the outside of the body like pain and temperature, that's also somatic nervous system. But if you needed to talk to cardiac and smooth muscle, as well as a lot of glands in your body, remember that's part of the nervous system you don't control, which is autonomic nervous system. So when you think about everything the nervous system does, it's really just 3 basic things. So we're going to boil it down to this. I want you to know there's 3 basic functions of the nervous system. And we've kind of mentioned at least 2 of them. We already know our nervous system is going to help us to sense our environment. So a sensory function is a function of the nervous system. It's helping you to sense your environment. We saw that another function of your nervous system is to talk to muscles, to stimulate muscles for movement. When you stimulate muscles for movement, we call that the motor function. I kind of think you move with motor. And then one last thing your nervous system does is that it processes information. It analyzes information. We call that the integrative function of your nervous system. So as we talk about all the different things your nervous system does, it's just these three basic things: sensory, sensing changes in environment; motor, stimulating muscles for movement; and integrative function, which is just you processing information, almost like a computer kind of thing. When you're taking your exam, you're processing information. You're using a lot of your integrative function of your nervous system. So as we go through this chapter, like usual, we're going to break it down both in terms of anatomy and physiology. And like usual, we're going to look at the anatomy both macroscopically, looking at structures we can see with our eyeballs, like the brain and spinal cord. But we're also going to have to look at it microscopically, meaning tissues. Remember, I told you, a lot of these tissues are going to come back throughout the semester as we hop from organ system to organ system. So the first thing we're going to do is remind ourselves about nervous tissue. Think back, we did this already. Remember when we looked at nervous tissue, you saw really two major groups of cells. Kind of imagine, we saw the bigger looking cell was the neuron. And you remember the neuron is doing most of the work. And then around the neuron, kind of imagine, if you can remember, we saw little speckled looking cells, little round cells. Those were your neuroglial cells. But this is lecture. We're going to focus in on each of those cells and go into a little bit more detail now, because we're breaking down the histology once more with a little bit more detail. So we're going to start off first with the neuron. Remember, the neuron is doing most of the work. And the way it's doing the work is actually by making electricity. A lot of the communication that occurs between neurons is literally little sparks of electricity. And so, in order for them to communicate, they actually make it. And when a cell can make electricity, usually in response to a stimulus, we say the cell is electrically excitable, meaning it can make electricity. So we're going to slow down a bit and kind of go over the anatomy, the basic anatomy of a neuron. And when we look at a neuron, we break it down into three basic parts. There's the body, sometimes called the soma, S-O-M-A. There's the axon and the dendrite. I want you to know the three parts of a neuron. And really, what are they doing? So let's go one by one, starting off with the body, or what we call the soma. You could always locate the body, the cell body, because it's where you're going to find most of the organelles, including the nucleus. So when we look at this image of this neuron here, you kind of use the cartoon. It's a yellowish looking neuron. And right away, you can see that round cluster called the nucleus. So that whole globular portion of the neuron that contains the nucleus and other organelles is the cell body. That's typically where the neuron is going to do some processing of information. But for it to process information, it needs to receive information. Turns out the neuron is going to receive information from another part of its entire cell. It's going to receive information in what we call the dendrites. You can see dendrites as short projections coming off the cell body. And if you follow the arrows in the picture, they're really showing you the pathway for information. So you can see information via these black arrows are coming in through the dendrites, because that's the dendrite's job. It's to receive information. And then you'll process it in the soma, or the cell body. And then you're going to have to send out information. You're going to have to communicate as a cell. The cell is going to use this last part to communicate. It's this long projection you see off the cell body called the axon. The axon is going to be how the neuron sends out information. So those are the three basic parts of a neuron. Short projections off the cell body are called the dendrites. They're there to receive information. Long projection off the cell body is the axon. That's how it will send out information. And the large globular portion containing most of the organelles, including the nucleus, is the soma, or the cell body, where you're going to process information. There's a couple more things to see here on this anatomy of this basic neuron, particularly when we look at the axon. It's one long projection off the cell body. But when you look at the axon, you could see some things on it. Remember, this neuron in this cartoon looks kind of yellow. And when you look at the axon, you see this pink stuff kind of wrapped around the axon in these little globs. We name these kind of globular structures sitting on top of the axon. It's called the myelin sheath. If you use a different textbook, they'll just call it myelin. This is really this plasma membrane of a different cell that's been wrapped around the axon. And you remember, when you think about the plasma membrane, it's mainly just fat. Remember, it's mainly phospholipids, and phospholipids are fat. So you could kind of imagine myelin is just fat wrapped around the axon. We'll see why that happens later. Turns out it's going to help with some functionality of the neuron. Besides these little globs of myelin, we see some other things. You notice that the myelin is not covering the entire axon. There are some places where the axon is exposed, where there is no myelin. We actually name these areas. The areas that do not have myelin are what we call nodes of Ranvier, while the areas that do have myelin are called internodes. So an internode is just a segment of axon that has myelin, and a node of Ranvier is just a segment of the axon that does not have myelin. Two more things to see here on this picture. If you notice, kind of closer to the cell body, still looking at the axon, just closer to the cell body, you notice that there looks like there's a little branch off the axon. We call that an axon collateral. Your axons could sometimes branch, kind of like a tree. And we call the branches axon collaterals. They're still just projections of the axon. Nothing major to know about them. And then if you follow the axon all the way to the end, it comes to these little globular tipped ends. We call them the axon terminals. They kind of tell you the name. Axon terminal. They're the ends to the axon. That's usually where your neuron will talk to another cell from using their axons, or at least the axon terminal of their axon. So that's the basic neuron anatomy. And when we look at the body, we notice that we have lots of neurons. Remember, this is a tissue. So neurons are cells. At one level, you are nothing more than a lot of cells. And when it comes to neurons, not all neurons look the same. And there are a lot of them. So you know what's going to happen. We're going to have to classify them. And when we look at neurons, one way to classify them is structurally. I want you to know the 3 basic structural classes of your neuron, and I want you to know, what does the neuron look like. This is a structural class, so you got to know, what does the neuron look like. So we'll go one by one. There's 3 of them. There's a multipolar neuron, a bipolar neuron, and a unipolar neuron. If you use a different textbook, they sometimes call the unipolar neuron a pseudo-unipolar neuron. So let's go one by one. Starting off with a multipolar neuron. By definition, a multipolar neuron has one axon and multiple dendrites. That's all it means to be a multipolar neuron. You have one axon and multiple dendrites. As you can see in the picture, one axon and multiple dendrites. Then there's a bipolar neuron. A bipolar neuron, by definition, has one axon and one dendrite. In the picture, that middle neuron is a bipolar neuron. You're seeing one axon on one side of the cell body and one dendrite. Dendrites can branch, but that is still just one dendrite. That is a bipolar neuron. Then the last type of neuron, structural class, is what we call a unipolar or pseudo-unipolar neuron. This one's kind of tricky. When you look at the image, it looks like there is no real axon or dendrite. It's because it's not. By definition, a unipolar neuron has the axon and dendrite fuse into one long extension. We call these extensions processes. So in a unipolar neuron, the axon and dendrites fuse into one long process or one long extension. So those are your structural classes. One more time. You have one axon and multiple dendrites? Well, you're a multipolar. One axon and one dendrite? Bipolar. Or did the axon and dendrites fuse together into one extension? A unipolar or a pseudo-unipolar. And it turns out most of the neurons in your body are multipolar neurons. So if you were to pick out a neuron out of your body, odds are you're just going to see one axon and multiple dendrites, because most of your neurons are multipolar neurons. Those are your structural classes. But in this class, if you could classify something structurally, well, then we're going to be able to classify it functionally. And remember, there are lots of different neurons in your body. We're only sticking to the ones in your exam review, only sticking to the ones we're mentioning. For example, there are other neurons like Purkinje cells and pyramidal cells, but you do not need to know those. So only stick to the ones we're talking about. So now we're going on to the functional classes. Turns out there are also 3 functional classes of neurons. Again, I want you to know these 3 classes, and I want you to know now, in terms of function, what are these neurons doing? So there are 3 functional classes. There are sensory neurons, sometimes called afferent neurons. There are motor neurons, sometimes called efferent neurons. And there are association neurons, sometimes called integration neurons. So what's going on? First up is the sensory neuron, sometimes called afferent. And by the way, afferent processes, or the term afferent, means to come towards the body, or in this case, towards the brain. While efferent means to go away from the brain, or away from the body. So just FYI. So first up is a sensory neuron, or an afferent neuron. Like usual, it tells you the name. It's a sensory neuron because it's doing the sensory function. It's helping you to sense your environment. And so you can imagine this is an afferent process. Kind of imagine if you were to touch something cold, you have to take that cold information from the neurons and the nerves of your fingers, and you're going to have to send them to your brain to let you know this is cold. That's an afferent process. So we call them afferent neurons because they send this sensory information towards the brain. Then there are motor neurons, sometimes called efferent neurons. And again, they tell you the name. Motor neurons are doing the motor function of your nervous system. They're talking to muscles. They're stimulating muscles. And again, you got to imagine, for me to stimulate muscles like walking, it actually begins in my brain. If I wanted to walk, it begins with neurons in my brain. That will then talk to neurons in my spinal cord. That will then talk to neurons in my nerves. That will talk to muscles. So this is going to begin in my brain. I'm going to have to send that information away from my brain towards my muscles. That's an efferent process. That's why we call them efferent neurons. And then the last group of neurons are called interneurons, or association neurons. They're doing that last function of the nervous system, which is the integrative function. They're just processing information. So those are the 3 functional classes. Turns out they correspond to the basic 3 functions of your nervous system. Are you sensing changes in the environment, doing the sensory function? Well, that's a sensory or afferent neuron. Are you stimulating muscles for movement? Are you doing the motor function? Well, that's a motor or efferent neuron. Or are you processing information? Are you doing the integrative function? Well, you're an interneuron, sometimes called an association neuron. Those are your 3 functional classes. And like usual, the structural and functional classes are related. And you got to know how. So let's remind ourselves about the 3 functional classes. Remember, there is 3 structural classes. There's a multipolar neuron, a bipolar neuron, and a unipolar neuron. Turns out the multipolars I mentioned earlier were the most common neuron in your body. They're so common, they're actually doing 2 of the 3 major functions. Turns out your multipolar neurons are helping you to talk to muscles. They're doing the motor function, and they're processing information. They're doing the integrative function. So multipolar neurons structurally are both motor neurons and interneurons functionally. And then that leaves the last 2 groups of structural neurons. There's the bipolar and the pseudo-unipolar or unipolar. They are both sensory neurons. Bipolar neurons and unipolar neurons are sensing changes in the environment. Turns out bipolar neurons are helping you with special senses like smelling, hearing, vision, while your pseudo-unipolar neurons are doing more of the basic sensations like pain, temperature, and light touch. So those are your functional and structural classes of neurons. And again, remember, there are lots of other types of neurons doing lots of things. Only stick to the ones we mentioned. For example, on this slide, you notice that there are several different types of sensory neurons. A sensory receptor is a specific type of neuron. One example is a nociceptor. Their specific sensation is to sense pain. So whenever you're walking around your house barefoot and you hit your pinky toe on the side table and you almost die, well, you feel the pain thanks to the nociceptors in your pinky toe. They're sensing the pain. But again, you don't need to know this. Just know the three structural and functional classes and how they're related. Remember, trust the exam review. And then lastly is the other cell in this tissue. Remember, the big globular cells doing most of the work with the neurons. And then when we looked under the microscope, we saw small little round cells. Remember, those are what we call the neuroglial cells. Remember, they are not doing the major work of the nervous system. Why? Because they're not electrically excitable, meaning they don't make electricity like the neuron does. So they're not sending and receiving signals. So what are they doing? Well, it depends on the cell. Turns out we never identified each individual neuroglial cell under the microscope because it's too hard at that level. But we do know the different neuroglial cells and what are they doing. Turns out there are 6 major types of neuroglial cells in the body. I want you to know your 6 neuroglial cells. Where do you find them? And by where, it's either central nervous system or peripheral. And what's their specific function? So like usual, we're going to slow down and go one by one. And we're going to first start off with the neuroglial cells in the central nervous system. Turns out there are 4 of them. The 4 cells in the central nervous system are the astrocytes, the oligodendrocytes, the microglial cell, and the ependymal cell. And you see them all represented in this cartoon. I'll slow down and go one by one. Just want you to know the cell, know the location, and know the function. And so these first 4 cells, you already know the location. They are all in the central nervous system. They are all either in the brain and or spinal cord. So let's go one by one. Starting off with what we call an astrocyte. In the cartoon, the astrocytes are the blue cells you see in the picture. And you can see these blue cells tend to only seem to be touching 2 things. Some of these cells seem to be touching blood vessels, and some of these cells seem to be touching the large yellow neurons. Oh, why is it? Why are they touching neurons and blood vessels? It's almost like they're separating them. It's because they are. Yeah. The function of the astrocyte is to form what we call the blood-brain barrier. Astrocytes form the blood-brain barrier. And it tells you in the name. It's literally a barrier between the blood and nervous tissue. Why? Because you would think tissue made up of cells would need oxygen and nutrients, which they'll get from the blood, which is true. But sometimes you get things in your blood that you don't want getting to your tissues. I'm talking about infections. Sometimes people get infections in the blood. It's called sepsis. And you don't want that to spread to the brain and make your brain or spinal cord become infected. So your body actually has a natural barrier, almost like a little filter between the blood and brain and spinal cord tissue. We call it the blood-brain barrier. That's why you don't really hear people talk about, oh man, I had the worst brain cold yesterday. It doesn't happen. Why? Because you have a blood-brain barrier made up of astrocytes. It's there for protection. So that's it. Keep it that simple. Astrocytes form the blood-brain barrier to protect brain tissue and spinal cord tissue from possible pathogens in the blood. Let's go on to another cell. Another cell in your central nervous system is called the microglial cell. And in the cartoon, the microglial cells are the little green cells you see in the picture. They're not really touching anything. They're kind of just hanging around. Why? What are the job of the microglial cells? Well, technically, microglial cells are basically white blood cells. Remember, you have white blood cells scattered throughout your body in different tissues. Remember, in skin, we saw white blood cells as the Langerhans or dendritic cells in the skin, helping to fight infections. Well, sometimes you might get an infection or a pathogen, a germ in your brain or spinal cord tissue, and you're going to need white blood cells to fight it. That is the microglial cell. Their job is to fight infection and remove debris, remove garbage, remove whatever doesn't belong from this tissue. That's the microglial cell. They fight infections and remove debris. Two more cells here. Another group of cells in the central nervous system are what we call ependymal cells. Ependymal cells. On the cartoon, they're the sheet of cells towards the bottom. That's your ependymal cells. And they really do two major things. One thing the ependymal cells do is they line cavities. They line empty pockets in the brain and spinal cord. Remember, there are empty pockets, or you'll see, there are empty pockets in the brain and spinal cord, and they'll be lined with this sheet of cells called ependymal cells. And these cells actually also make a fluid. I want you to know the fluid your ependymal cells make. It's called cerebrospinal fluid, sometimes abbreviated CSF. Cerebrospinal fluid. That's the fluid that's kind of buoyant within and around the brain and spinal cord. Why? Well, you could kind of think your brain's sitting in your skull, and we've seen skull bones are hard, and your brain has the consistency almost like pudding. So if you were to leave the brain just sitting on your skull bone inside your head, it would basically collapse under its own weight. But luckily, your brain is floating in your skull, and it's floating in cerebrospinal fluid created by your ependymal cells. So keep it this simple. Ependymal cells line empty cavities and make cerebrospinal fluid for the brain and spinal cord. And then there's one more cell here in the central nervous system. It's called the oligodendrocyte. It's the pinkish looking cell in the picture. Almost look like octopi in the picture. If you notice, they look like they're sending these long extensions and wrapping them around the axon. Oh, that looks almost like the pink stuff we saw in the picture of the neuron earlier. It almost looks like myelin. It's because it is. The job of the oligodendrocyte is to make myelin in the central nervous system. And you can see from the picture, which is true for real life, oligodendrocytes can make myelin from more than one axon, from more than one neuron. So those are the neuroglial cells in the central nervous system. Astrocytes forming the blood-brain barrier, microglial cells fighting infections and removing debris or garbage, ependymal cells lining empty cavities and making cerebrospinal fluid, and oligodendrocytes making myelin from multiple axons. That's it. Keep it simple. Know the cell, know the function, and these four are all located in the central nervous system, brain and spinal cord. Then to finish off your neuroglial cells, we look at two more. We look at two of them located in the peripheral nervous system, located in your nerves. There's the satellite cell and the Schwann cell. Those are the two neuroglial cells in your peripheral nervous system, satellite cell and the Schwann cell. And we'll do the satellite cell first. And you can see it on this picture all the way to the right, labeled C. When you look at the soma, or the cell body of the neuron on this image, you see all these cells kind of sitting and hovering around the soma, almost like satellites kind of hover or surround the planet. That's why they're called satellite cells. They look like they're hovering around, orbiting around the soma, almost like satellites orbit around the planet. But what's the function? Remember, you got to know the cell, the function, and the location. And satellite cells are a little tricky. We know they're helping the neurons, but we're not quite sure how. The research isn't quite there yet. So if I were to ask, what's the function of a satellite cell? It's just to support a neuron. And if I were to ask how it's supporting a neuron, well, the true answer is I don't know, because science isn't there yet. So keep it this simple. Satellite cells, for now, we just know, support the neuron. How? Not quite sure. And then the last cell is what we call a Schwann cell. Schwann cell. You can see it in images A and B on this label A and B on this image. And again, they look pink, wrapped around the axon. It looks like myelin because it is. Schwann cells also make myelin, but they only make myelin for the peripheral nervous system. So don't get these two cells confused. Oligodendrocytes make myelin for the central nervous system, brain and spinal cord, while Schwann cells make myelin for the peripheral nervous system, for your nerves. And we see one more difference. Remember, oligodendrocytes can make myelin for more than one neuron, more than one axon. But one Schwann cell could only make myelin for only one axon. So those are your cells. Know the cell, know the location, and know the function. Keep it as simple as we've been describing it. And we saw your cells, your neurons, have at least 2 neuroglial cells dedicated to just making myelin. So we got to look at that. How does it occur? How do they make the myelin? I mentioned earlier when we looked at the anatomy of a neuron that myelin is really plasma membranes of these cells. It's really fat. And all these cells literally do is they sit on the axon and they stretch out their plasma membrane and wrap it around the axon multiple times. It's literally majorly fat. That's all myelin is. And the process of making myelin is what we call myelination. And so Schwann cells and oligodendrocytes do myelination. They make myelin. And I told you, it's just fat. And we saw your neurons don't necessarily have myelin covering the entire axon. And in fact, not every neuron actually has myelin. And we could see that visibly with just the naked eye. For example, when we look at the brain and spinal cord, we break them up into areas, mainly different colored looking tissues, what we call gray and white matter. When you look at gray and white matter, you're just looking at where there may or may not be myelin on the axons of the neurons in the tissue when you're looking at these organs. First is the spinal cord, which is what you see labeled A on the image to the left. The spinal cord, when you look at it more on the outer portions, more on the periphery, more superficially, it looks lighter. That's white matter. And when you look at white matter, you're looking at fat on the neurons. You're looking at where there are myelinated neurons. You're looking at where neurons have myelin. But if there is no myelin, you're going to be looking directly at the neurons, which looks a little darker. Kind of, for example, when you look at the central portions of the spinal cord, it looks darker. That's where we have gray matter. Gray matter is where you're seeing unmyelinated neurons. It's where you're seeing neurons that do not have myelin. And it turns out myelin helps neurons to send information a little bit faster. We'll see how a little later in this chapter. So right off the bat, you know something right away about the spinal cord, based off gray and white matter, based off where you do or do not have myelin. The white matter, more on the periphery of your spinal cord, is myelinated, has neurons with myelin, so they're sending information faster than the central portions of your spinal cord, which has gray matter, which has unmyelinated neurons, which has neurons that do not have myelin. And same thing kind of occurs for the brain. It's just that the locations are a little different. On the image to the right is a coronal section of the brain. And you can see the more periphery now looks darker because that's where the gray matter is. It's more superficial, or more in the periphery in your brain, while the central portions of the brain are more white matter. So you can see the central portions of the brain are likely to send information faster than the superficial portions because the central portions have myelinated neurons. So that's the difference. Gray matter, neurons do not have myelin. White matter, neurons have myelin. And that's the basic histological anatomy. That's looking at things under the microscope. Now we're going to switch gears and talk about some physiology for all this microscopic stuff. So we know what neurons are doing. We know what neuroglial cells are doing. But now we got to talk about how. And we're going to focus in on the neurons. We know they're communicating with electricity. So we're going to have to talk about that. Turns out your neurons, we already know, can make electricity. They're electrically excitable cells. And when they make sparks of electricity, we got to talk about that. Electricity is really moving charged particles, things with a positive or negative charge. When they move, we call that electricity. But when we talk about electricity and neurons, we give a name to it. There are multiple names for this. These sparks of electricity could be called action potentials or graded potentials. So whenever you see the terms action potential or graded potential, we're just talking about sparks of electricity. So what's the difference? Why do we have different names for a spark of electricity? Well, it has to deal with how far that electricity could move. If you make a spark of electricity that could travel a long distance, we could call that an action potential. Usually, this is a spark of electricity that could travel the length of an axon. Remember, you're going to send information out via the axon. And now you know the information you're sending out is in the form of electricity. And if that electricity travels a long distance, aka the full length of an axon, that is an action potential. But your spark doesn't necessarily have to go very far. Your spark of electricity could go a short distance, not the full length of an axon. If it goes a short distance, we call that a graded potential. So that's the major difference. Action potentials and graded potentials are both sparks of electricity. It's just that the action potentials could travel a long distance, while a graded potential travels a short distance. So we're going to have to talk about this. How do you make an action potential? How do you make a graded potential? And how is that going to lead to you doing all the functions of the nervous system? Like things like talking to muscles to help you to take your notes. So let's get into it. And it actually has to do with the plasma membrane of the neuron. So before we even dive in, we got to remind ourselves about plasma membrane anatomy. That's what you see in the bottom images down below. You're just seeing a segment of the plasma membrane. You remember your plasma membrane is a phospholipid bilayer, meaning you're seeing two layers of phospholipids. But you remember there are also other things in the plasma membrane, like proteins that embed themselves into the membrane. Remember, we call those integral proteins or transmembrane proteins. And I told you a lot of these proteins are acting as channels to let things in and out of the cell. When it comes to making this spark of electricity, these integral proteins are going to play a huge role in helping to make this spark, helping you to move charged particles. So we're going to first talk about these integral proteins, these protein channels that are going to help you to move charged particles like positively charged sodium or positively charged potassium. So let's go through them. First up is what we call a leaky channel, sometimes called a leakage channel. They kind of give you a name in the hint. They're leaky because they're always on. I know in your slide it says they alternate between open and closed. That's not really what's the case. The case is they're usually open, kind of like a leak. It's open, so it's leaking. These are channels that are all open and allowing things to leak either in or out of the cell. So this is going to be one way you're going to be able to move charged particles. But they're not the most common channel involved in making action and graded potentials. The most common kinds are channels that are usually closed and you're going to have to do something to open them. Channels are these integral proteins that are closed and you need to do something to open them are called gated channels. Kind of thing. It's like you have a gate. To get in, you got to open the gate. You have to open these channels. And we name these channels based off what you're going to use to open them. For example, some of these channels, some of these integral proteins will only open when you have a special chemical stimulus. We call these chemical stimuli ligands. A ligand is just a chemical that could open the gate. So if you're a channel that only opens when there's a ligand, we call you a ligand-gated channel, which is what you see in this image. As soon as the ligand binds, the channel will open. That's a ligand-gated channel. It's usually closed, and you need a ligand to open it. Other channels open with other stimuli, like this next one here. It's called a mechanically gated channel. These will open if you apply some type of mechanical stimulus. And whenever you hear the word mechanical in anatomy, think physical. So you have to do something physical to it, like maybe shake or vibrate the channel, or squeeze and apply pressure, or push it. Do something physical or mechanical to open the channel. You're dealing with a mechanically gated channel. Keep going. You'll see one channel that plays the largest role in helping you to make your action and graded potentials are what we call voltage gated channels. Again, these are gated channels, so they're usually closed. And in this case, to open them, you're going to have to change the voltage across the plasma membrane. Oh, what's that? What's the voltage? In this case, the voltage is what we call the membrane potential. Oh, that didn't help either. What's the membrane potential? The membrane potential, by definition, is just a difference in charge across the membrane. That's all it is. A difference in charge, either negative or positive. If you change the charge, aka the membrane potential, aka the voltage of your plasma membrane, that will open the channel. So these are your different channels. These are different integral proteins, and they're going to have a role to play in helping you to make your spark of electricity, either your action or your graded potential. And this is going to either lead to you sensing changes in your environment, talking or stimulating muscles, or processing information. So let's do an example. Let's say you wanted to walk to the door. It's going to begin with neurons in your brain that will need to make sparks of electricity. And before you move, these cells are resting. When you're not talking to muscles, when you're not stimulating muscles, your neurons are not doing anything. They're resting. And if you were to check the difference in charge across the membrane when they're resting, we give that a name. This difference in charge is across the membrane when a neuron is at rest is what we call a resting membrane potential. It is literally just the difference in charge across the membrane. And we know the difference. When we look at a neuron, when it's resting, when we look at a neuron's resting membrane potential, it's more positive along the outside of the plasma membrane and more negative along the inside, when you're literally not doing anything. But even though you're not doing anything, the neuron isn't doing anything, those integral proteins are working. And they're moving charged particles. They're moving those two charged particles I mentioned before, sodium, positively charged sodium, and positively charged potassium. And when these ions, these charged particles move, they're going to create this difference in charge across the membrane, where you're going to be more negative on the inside and more positive on the outside, which is what you can see on the image here. Over to the right, you literally see a little small voltometer, which is measuring volts, because we're dealing with electricity. So we use units of measurements for electricity, which is volts. And you could literally measure it. And we see that usually when your neuron is at rest, it's going to be slightly negative along the inside of the plasma membrane. And along the outside, facing the external environment, it's going to be slightly positive. This difference is what we call the resting membrane potential. And we can measure this difference. I want you to know, what is the resting membrane potential for a typical neuron? Again, you can see it in the image to the right in the little voltometer. They're measuring it. That average resting membrane potential for your neurons is about negative 70 millivolts. Again, just break it all down. Look at the units of measurements. We're dealing with electricity, so the units are volts for voltage. But again, you got to think, this is a cell. This is at the cellular level. These are tiny changes in voltage. So we're talking about millivolts. And then it's negative 70. That negative 70 is saying the cell is about 70 times more negative on the inside of its plasma membrane than it is on the outside. That is all your resting membrane potential is. This is the baseline of where your neurons are going to be, where they're not doing anything. And so when we're going to make our spark of electricity, we're going to move charged particles, and that will end up changing the membrane potential. You're going to change this voltage, this difference in charges across the membrane. and that will lead to you making your spark of electricity. And you're going to be able to move these charged particles with all those different types of integral proteins, all those different types of protein channels embedded in the plasma membrane. And remember, it helps to just think. We're just talking about electricity, little sparks of electricity. And again, I remember I mentioned there's two major types. There are the action potentials that travel the full length of the axon, go very long distances, and there are graded potentials, which just happen to go short distances. So they're not going to make it all the way down the axon to the axon terminal. And again, remember, stick to the exam review. I'm going to talk about graded potentials, but I'm not really going to test you on it. Stick to the exam review. I'm just going to go through these next couple of slides over graded potentials just for thoroughness. Just bear that in mind. And so again, a graded potential is basically a spark of electricity that's generated thanks to changes in that resting membrane potential, thanks to changes in the difference in charges across the membrane. And when we talk about these changes in charges across the membrane, they're not great changes. These are small deviations from the resting membrane potential, meaning it's just going to be a small change from that average negative 70 millivolts resting membrane potential that we are going to see in our neurons. And again, just like an action potential, these graded potentials are generated thanks to these changes created by opening and closing all those different types of membrane channels, those integral proteins embedded in the membrane. And again, stick to the exam review. But there are some other differences between action potentials and graded potentials besides how far they could travel. For one, a graded potential can increase in strength over time. It just depends on the stimulus. What you're seeing in this chart is kind of imagine pricking your finger with a little pin one time. That's probably going to cause some painful sensation, but it should dissipate rather quickly. That's because the stimulus in that small pinprick isn't that big. It's like stimulus 1. It's going to create just a small deviation from that resting membrane potential. You're going to change the charge just a bit. But let's say instead of just getting pricked with a pin, maybe you're cooking dinner and you actually slice through your finger. That's going to cause a bigger stimulus for your body to pick up. And you're going to get a bigger amplitude in your graded potential. Basically, think a bigger spark of electricity. Because you got to think, now your body needs to tell you that you're in more pain. So it needs a bigger stimulus and a bigger potential to help send that signal to the brain. And you can imagine stimulus 3, that's probably you losing a limb. You're going to, again, if you lose a limb, that's going to be a big pain signal. So you're going to have a big stimulus. You're going to have to tell the brain now, I'm in a lot of pain. So you're going to have to generate an even bigger graded potential. So this slide is just basically showing that graded potentials can vary in size. It's just related to the stimulus. What's another difference? Turns out not only do graded potentials differ in size, they could actually combine together. It's like getting two small signals that you're trying to send to or away from the brain and combining them into a larger signal. Again, stick to the exam review. You could skip over this slide. What we want to focus on is the action potential. Again, imagine it as a spark of electricity your neuron is creating, and it's sending it along the full length of the axon because it could travel a long distance. And how do you create it again? You just change the difference in charge across the membrane. You change the resting membrane potential. That's all that's happening when you have an action potential. It's just a series of events that change that charge across the membrane potential, and that will result in this spark of electricity we call an action potential. What we want to focus on is how do you create this? How do you create this spark of electricity called an action potential? How do we change this charge across the membrane? You know something about it. It has to deal with those integral proteins. But we're going to make it simple. We're going to break down how do you create an action potential into 4 major steps. I want you to know the 4 major steps in the generation of an action potential. First, I'm going to give you the names of the steps, and then we'll go one by one talking about what's happening. Make sure you know them in order and what happens in each step. So the first step is called reaching threshold. Second step is called depolarization. Third step is called repolarization. And the fourth step is called hyperpolarization. This chart here on slide 35 is just showing us all four of those major steps. Know them in order and what happens in each step. Okay, so let's go back over these steps one by one in order to figure out what's going on in each step. Starting off with step one, which is called reaching threshold. Well, you know that in order to make this action potential, this spark of electricity, we need to open some of these gated channels. And the channels that are mainly going to open, you can see from this slide, are voltage gated channels. And remember, these voltage gated channels only open when you change the voltage, when you change the charge across the membrane. And we actually know how much you need to change the charge from the resting membrane potential. It's in the chart. Turns out you need to hit about negative 55 millivolts. Once you hit negative 55 millivolts, that's called hitting threshold. And the way you're going to do that is thanks to some somewhat leaky or somewhat open sodium channels that are not voltage gated. But keep it this simple. First step is to hit negative 55 millivolts. We call that reaching threshold. That's it. Then once you hit threshold, you're going to move on to step two, which is called depolarization. So what's happening here? It's this green segment of the line on this line graph you see on the slide. You can see in this area of that green, which is depolarization, it's going from about negative 55 millivolts all the way up towards the positive end, all the way up towards about positive 30 millivolts. This is depolarization. It's when the cell becomes more positive on the inside. Remember, when the cell was resting, when we looked on the inside of the plasma membrane, it has a slightly negative charge. When that negative charge becomes positive, that is depolarization. And how do you do that? How do you depolarize? Remember, you're going to have to open a channel. Turns out the channel we're going to open in depolarization are sodium channels, voltage-gated sodium channels. They'll open up. Sodium will rush into the cell. Positively charged sodium will rush into the cell, and it will bring its positive charges with it to make the cell positive on the inside. Again, keep it simple. Depolarization is how the cell becomes positive on the inside of its plasma membrane by opening voltage-gated sodium channels and allowing sodium to rush into the cell. And the cell will become positive along the inside of its plasma membrane. And that pretty much makes the spark called the action potential. But you remember, there are two more steps. There are 4 steps total. Think of these last 2 steps of kind of your neuron resetting. It needs to reset so that it could get ready to send another signal. So think of these last 2 steps as almost like your neurons resetting so it could do this same thing over again. So the third step I mentioned earlier is called repolarization. And on your slide, on the graph, it's represented by the little red line. Repolarization, you can see from the red line, now it's going back from about positive 30 millivolts all the way back down to the negative range, almost back to negative 70. This is what happens in repolarization. In repolarization, the cell goes back to being negative on the inside. It kind of tells you in the name, to repolarize. Because in the beginning, you remember at the resting membrane potential, you were positive on the outside, negative on the inside. It's almost like you had 2 poles or 2 ends, like a battery, a positive and negative end. And when you depolarized, you made that negative side become positive. So it's like you lost your polarity. Well, now you're going to bring it back. Now the cell is going to go back to being negative on the inside. That's repolarization. And again, it has something to do with those channels. So how do you repolarize? How do you go back to being negative along the inside of the plasma membrane? Well, for one, you got to stop letting positive stuff in. Remember, you were letting in sodium in depolarization. So you got to stop that. You got to close sodium channels and stop letting sodium in. But that's not enough. You got to do one more thing. You're going to now open voltage-gated potassium channels. and you're going to kick out potassium, positively charged potassium. You're going to kick it out of the cell, and it's going to take its positive charges with it, and the cell's going to go back to being negative on the inside. That's repolarization. Again, keeping it simple, repolarization is when the cell becomes negative along the inside. How? You're going to close sodium channels. You're going to open voltage-gated potassium channels and kick potassium out of the cell, and then the cell will go back to being negative on the inside. And then there's one last step. It's called hyperpolarization. It's like sometimes your neurons kind of do too much of the repolarization phase. Hyperpolarization is when the cell becomes more negative than it was at rest. When the cell becomes more negative than its resting membrane potential. That's the brown part of the line you see on the graph. You can see it's gone past the negative 70, almost all the way to negative 80 millivolts. Hyperpolarization is when the cell becomes more negative than it was on the inside. And that's all you have to know about repolarization. There's a little bit more that goes along with it, but I will not ask. Your potassium channels will close, so you'll stop kicking out potassium, so you won't become even more negative. And your leaky channels, those channels that are always open, those will kind of get you back to your resting membrane potential. But you don't need to go any further. So again, to summarize, the 4 steps of making an action potential, make sure you know them in order and what happens in each step. First step is called hitting threshold. That's when the cell's resting membrane potential changes from about negative 70 millivolts to negative 55 millivolts. Then you'll depolarize, becoming more positive on the inside of the plasma membrane by opening voltage-gated sodium channels and allowing positively charged sodium to rush into the cell. Then you'll repolarize, become negative along the inside of the plasma membrane by closing your sodium channels and opening voltage-gated potassium channels and kicking out positively charged potassium. And then you'll finish off with hyperpolarization, or you'll become more negative than your negative 70 millivolts, more negative than your resting membrane potential. And that's how you'll make a spark of electricity we call an action potential. Keep it that simple. Now that you know how to make an action potential, we could talk about a little bit more interesting facts about an action potential. Remember, there are some differences between this and a graded potential. One is how far can they travel. Another difference was that we saw graded potentials could vary in size. Turns out action potentials cannot vary in size. You could kind of think it's one size fits all. You make the same size action potential every time. You either make it or you don't at all. There's no in between. And so it's very important to do that first step of making the action potential. It's very important to reach threshold, to reach that negative 55 millivolts. Because if you don't, well, you're not going to make the action potential because it needs to be long enough. It needs to be big enough to make that long travel down the axon. So again, make sure you know your steps in the generation of an action potential. Hitting threshold, depolarization, repolarization, and then you finish off with hyperpolarization. This chart, table 12.2, is just again giving some more differences between graded and action potentials if you're curious. But again, stick to the exam review. And you remember, we're talking about this in order to get through the physiology of a neuron. We're really talking about how do neurons send information, receive information, and process it. And it's doing it with these sparks of electricity. That's why we're talking about them. And I told you this major spark that's doing a lot of the work for the neuron, or at least allowing the neuron to do the work, is the action potential because it's making it the full length of the axon. So we got to talk about that. Now you know how to make an action potential. How do you move it down the axon? How do you move electricity, basically? Well, normally, we know something from our everyday life. Think lightning. Lightning's electricity. And usually when something is able to attract lightning, we could say you could conduct it. Because that's really how you move electricity. It's conducting electricity. And where these action potentials are electricity. So when we talk about moving action potentials, we're really talking about conduction, moving electricity. And like usual, this is the body. You're going to have multiple options. And when it comes to moving the axon down the when it comes to moving the action potential down the axon, there are two major ways to do it. I want you to know the two major types of conduction for action potentials. There's something called continuous conduction and something called saltatory conduction. I want you to know the difference. I'm going to go one by one. They're both shown here on this slide. Over onto the left, labeled on the A column, that's continuous conduction. And over on the B side, you see on the right side is the B side, is saltatory conduction. And right off the bat, when you compare the right side pictures to the left side pictures, you're looking at a neuron. And you're looking at the action potential travel down the axon of the neuron. But you notice a difference between the right side and the left side right away. If you look at the neurons on the right side, it looks like there's these little red boxes kind of circled around the axon. Oh, those aren't red boxes. Those are representations of myelin. Now, remember, some neurons have that fatty myelin wrapped around it, and I mentioned it's helping to send the action potential faster. It has something to do with conduction. Turns out in saltatory conduction is what you see in neurons that have myelin. Saltatory conduction occurs in myelinated neurons, while continuous conduction occurs in unmyelinated neurons. It occurs in neurons that do not have myelin. That's why when you look over at the image to the left, you do not see those little red boxes, because there is no myelin involved in continuous conduction. It occurs in unmyelinated neurons. So that's one difference, is who does which. Myelinated neurons do saltatory conduction. Unmyelinated neurons do continuous conduction. Then how do they occur? Let's go through saltatory conduction first. In saltatory conduction, you're still making the action potential. You could kind of imagine as you're going down the axon, you still have to do those 4 steps over and over again. Hitting threshold, depolarization, repolarization, and hyperpolarization. For every segment that you go through of the plasma membrane of the axon, you're having to do those 4 steps. Except for where there's myelin. Wherever there's myelin, you do not need to do those 4 steps. It's almost like your action potential jumps over the myelin. And you remember, we named those areas of the neuron that do not have myelin. Remember, they're called nodes of Ranvier. So in saltatory conduction, we say the action potential jumps from node of Ranvier to node of Ranvier. So that's actually going to help the action potential to arrive at the axon terminal faster, because it's not having to go through every single inch or millimeter of that axon. It just jumps from node of Ranvier to node of Ranvier. You could kind of imagine trying to get to school. Imagine driving to school in a car. If you did something like continuous conduction in your car driving, that'll be like you driving the full road. But if you were doing something similar to saltatory conduction, that would be like you jumping from intersection to intersection. You don't have to drive the full length of the road. You're hopping from intersection to intersection, almost like the action potential jumps from node of Ranvier to node of Ranvier. So you're going to get to the end or to your destination faster. So we say saltatory conduction is faster than continuous conduction. Why? The action potential jumps from node of Ranvier to node of Ranvier. This is different for continuous conduction. Again, it tells you in the name. It's called continuous because you have to continuously go through all 4 of those steps for every single part of the axon. Why? Because you don't have any myelin to jump over. So it has to hit threshold, depolarize, repolarize, and hyperpolarize for every single part of the axon in continuous conduction. And that's going to take a while. So continuous conduction goes really slow, or at least slower than saltatory conduction. So again, like usual, keep it simple. Saltatory conduction is faster. It occurs in myelinated neurons, and the action potential jumps from node of Ranvier to node of Ranvier. Keep it that simple. And in continuous conduction, you have to continuously do your 4 steps of action potential generation for the full length of the axon. You cannot skip any portion. And this will occur a little bit slower. And this is what happens in unmyelinated neurons. That's it. That's continuous and saltatory conduction. Now you know kind of how you're sending the spark of electricity, the action potential, down the axon. And now that we know how it occurs, we could complicate it a little bit. Like usual, a lot of things in your body can get influenced by other things. So there are some factors that have a say on how fast you could send your action potential. I want you to know these 3 factors that have a say or an influence on how fast you could send your action potential. We're talking about action potential propagation, meaning how fast can you move it. And it's just 3 things. One of them is the size of the axon the diameter. Another one is whether or not you have myelin. The amount of myelination is basically saying how much myelin do you have it. And temperature. And like usual, you know one of the answers, or at least 2 of the answers already. But you got to think, one of the answers way back from when we did chemistry. Way back from when we talked about chemical reactions. Remember, at one level, your chemical level, you're just a whole bunch of chemical reactions occurring. And you remember, chemical reactions have a speed, and speed could be affected by temperature. Remember, fast or high temperatures make reactions go fast, and low temperatures make reactions go slow. So think of moving or generating action potentials as a chemical reaction. You know the answer already. You already know a higher temperature will cause you to send the action potential faster, and a lower temperature will cause it to go slower, just like a chemical reaction. This makes things make sense. This is why whenever you twist your ankle and you're in pain, what do you put on a twisted ankle? Do you put ice on it, or do you put a hot towel? Oh, I hope you don't put a hot towel on it. You should put ice on it, because that ice is lowering the temperature to slow down reactions like the generation of an action potential. So your brain is not going to get the pain information as fast, and you're going to interpret that as having reduced pain. Now you know why you put ice on a swollen ankle, at least with the pain. So that's temperatures. One more time. High temperatures make you send the action potential faster. Low temperatures, you send it slower. What's another thing that could affect how fast you could send it? Remember, it's the amount of myelin, whether or not you have myelin. We know that answer from the last slide. You know that if you do have myelin, you're going to do saltatory conduction. So that's going to be faster. And if you don't have myelin, well, you're going to do continuous conduction. That's going to be slower. Basically, if you have myelin or a lot of myelin, you'll go fast. And if you don't have myelin or extremely little myelin, you'll go slow. That's simple. And then the last thing is the axon diameter. How big is the axon? And it's this simple. A big diameter will go faster, and a small diameter will go slower. Again, kind of imagine driving. You could imagine which type of road would you go faster on? Would you go faster on a 2-lane road? Or would you go faster on a 6-lane highway? Well, you're going to go faster on that 6-lane highway. The more room you have, the faster you could go. Same thing for electricity. Same thing for your action potential. Give it a bigger diameter, you'll send the action potential faster. Give it a small diameter, you'll send the action potential slower. So those are things that could affect your action potential. Diameter size, bigger, faster. Smaller, slower. Amount of myelin, a lot or present, faster. Little or absent, slower. And temperature, higher temperature, faster. Lower temperature, slower. So you're learning a lot about neurons now. Now you know how to make an action potential, how to move an action potential, and the things that could affect it. Now we could talk about the end result. What happens when that action potential arrives at the axon terminal? Well, it's going to allow the cell to communicate. So we got to talk about that. How do cells communicate? Well, cells communicate with other cells, which can be another neuron or another cell in your body altogether. And where they communicate, we give a name to it. Where a neuron will talk to another cell, which can be another neuron, is called a synapse. And like usual, there are different types in your body. There are electrical synapses, where you're sending electricity from cell to cell. Kind of think in your heart, where you have gap junctions. Your heart has to beat at one, so all the cells need to beat at one, and they get the signal via electricity. So in your heart, you have a lot of electrical synapses, really to help with synchronicity, so your heart cells beat as one. But there are other synapses. Another group are called chemical synapses. These are where cells are communicating with chemicals. We call these chemicals lots of times neurotransmitters. They're like chemical messengers. But we're going to focus in on 3 specific synapses. I want you to know these 3 specific synapses on this slide here. And they're all synapses that occur between neurons. They're basically places where neurons could talk to other neurons. Know these 3 synapses and the parts that occur here. So there's an axo-axonic synapse, axodendritic synapse, and an axosomatic synapse. So let's go through them one by one. What do they look like? And like usual, they tell you in the name. Starting with an axo-axonic synapse. It tells you in the name. It's called axo for axon and axonic for axon. This is basically where the axon of one neuron will communicate with the axon of another neuron. That's an axo-axonic. Think axon to axon. Another synapse is called an axodendritic synapse. Again, just break down the names. Axo for axon, dendritic for dendrite. This is where an axon of one neuron and the dendrite of another neuron will communicate. Think axon to dendrite. That's an axodendritic synapse. And the last type of synapse is called an axosomatic synapse. Like usual, break down the names. Axo for axons, somatic for soma. Remember, soma means cell body. So this is where basically the axon of one neuron will talk to the soma, or the cell body of another neuron. So these are just showing different examples of synapses, different examples where neurons could communicate with other cells, which can be other neurons. Again, like usual, keep it simple. Is it axon to axon? Axo-axonic. Axon to dendrite? Axodendritic. Or axon to soma, or cell body? Axosomatic. So those are your 3 different types of at least neuronal synapses where neurons could communicate. And it all begins with this axon terminal. The whole reason for generating an action potential was so that it could reach the axon terminal. Why? Because the axon terminal, you saw from the previous pictures, is involved in those axon-based synapses, how they're going to communicate. And it has 3 basic parts to it. Whenever you look at any synapse, axo-axonic, axodendritic, axosomatic, etc., any synapse will have 3 basic parts to it. I want you to know the 3 basic parts to a synapse, and you'll see it on this slide here. Well, one part is the end of the axon of the neuron sending the signal. Remember, the end of the axon is called the axon terminal. So one part to your synapse is the axon terminal of the neuron sending the signal. Then there's going to be the cell receiving the signal, and it's going to receive the signal along its plasma membrane. That's going to be the second part. It's going to be the plasma membrane of the cell receiving the signal. And when these cells communicate, they don't have to touch. So there's going to be a slight gap between the 2 cells, and we name the gap. And this gap is the 3rd part to your synapse. It's called the synaptic cleft. So you see all 3 parts on this picture. You see the axon terminal, the neuron sending the signal. You see the plasma membrane of the neuron receiving the signal. And you see the gap in between, the synaptic cleft. And again, they're going to communicate depending on the type of synapse, whether it might be with electricity, with an electrical synapse or with chemicals in a chemical synapse. And they're communicating in a chemical synapse with tiny chemicals, literally. And I told you, we call them neurotransmitters. Turns out when I get up to walk or I move a muscle, it begins with my neuron talking to my muscle cell with a chemical, with a neurotransmitter. And then when I want to stop moving, I got to get rid of that neurotransmitter. I got to get rid of that chemical messenger. I want you to know the 3 major ways your body will get rid of a chemical messenger. The 3 major ways your body will get rid of a neurotransmitter. And you see them all on this slide here. Again, like usual, I'll name the 3 ways and then we'll go over them. The 3 ways, well, first way to remove a neurotransmitter is something called diffusion. Diffusion, like diffusion and osmosis we talked about earlier in the semester. So, diffusion. Another type is called reuptake. Reuptake. And the third type is called enzymatic degradation. Let's go through them. Diffusion, reuptake, and enzymatic degradation. What are they? First up is diffusion. Remember, in diffusion, that's when solutes move from an area of high solute concentration to low. This is basically where the neurotransmitter is going to move from where there's a lot of it to where there's very little. And you can see from the picture, you have a lot of neurotransmitter in your synapse. So in diffusion, your neurotransmitter will leave the synapse. It diffuses away from the synapse. It's like it leaves that location. So if it leaves, well, you're not sending the signal and you'll relax. So that's one way. Have your neurotransmitter leave the synapse, diffuse away. Another way is called reuptake. Well, for your cells to communicate, the neurotransmitter has to be released from the axon terminal. But sometimes when your neuron releases the neurotransmitter, it could suck it back up. It's like it sends the signal and it says, nah, and it sucks the signal back. We call that reuptake. And the last way to get rid of your neurotransmitter is called enzymatic degradation. This is basically when an enzyme breaks down your neurotransmitter. And if you break it down, well, it's gone and you're not going to send the signal. So these are 3 ways to get rid of a neurotransmitter. One way, diffusion, have it leave or diffuse from the synapse. Reuptake, have the neuron that released it suck it back up into the axon terminal. Reuptake. Or have an enzyme break down the neurotransmitter. That's enzymatic degradation. Those are 3 major ways your body will get rid of a neurotransmitter. But if you don't get rid of the neurotransmitter, what happens next? Once the cell that's receiving the signal gets that chemical messenger, what happens? Well, like always, it depends. There's options. It depends on what you want to do. Sometimes your neurons will tell another cell to do something. It will excite them. Or your neuron could tell a cell to stop doing something. It could inhibit it. If a neuron is responsible for stimulating a cell, telling a cell to do something, when that cell it sends the signal to, the neurotransmitter to, receives it, it's going to generate its own spark of electricity. And in order to do that, it has to do some of the similar steps of changing its plasma membrane, which involves depolarization. So if a cell gets told to do something, if it gets excited, it's going to generate what we call an excitatory postsynaptic potential, meaning it's going to generate its own spark of electricity. But you could tell a cell to stop doing something. If you tell a cell to stop doing something, well, it's not going to generate an action potential. How? You're going to cause it to hyperpolarize. It's going to become so negative that it cannot become positive on the inside. And it's like you've frozen that cell. It's going to generate what we call a inhibitory postsynaptic potential. It's going to hyperpolarize and stop doing something. Again, I'm just mentioning this for thoroughness. Like always, stick to the exam review. And when it comes to a chemical messenger, you actually have several different types. Your body doesn't just pick one chemical to send all the different types of signals. You have a different neurotransmitter for lots of different occasions. And some neurotransmitters are excitatory, meaning they will generate excitatory postsynaptic potentials in the recipient cell, meaning it will tell the recipient cell to do something. Or you have neurotransmitters that will generate an inhibitory postsynaptic potential in the recipient cell. It will tell the cell that receives it to stop doing something. And not only do you have different types of neurotransmitters, you even have different types of receptors for each neurotransmitter. And you don't need to know them. I just want you to know one particular neurotransmitter up close. It's called acetylcholine. Acetylcholine is a major neurotransmitter our body uses to help us to communicate with other cells, especially muscle cells, which is what we'll see in the next chapter. You spell acetylcholine ACetylcholine, acetylcholine. These next slides are just going over different types of receptors for neurotransmitters. You could skip over those slides if you want. And again, we've already talked about the 3 ways in which you remove a neurotransmitter. Just one more time for completion. Diffusion is when the neurotransmitter diffuses or leaves the synapse. Enzymatic degradation is when an enzyme breaks down your neurotransmitter. And reuptake, also called uptake, is when the neuron that released the neurotransmitter sucks it back up. It takes it back. It reuptakes it. Okay. So now you know a lot about how neurons communicate. You know how they're going to make their spark of electricity, where that electricity is going to go and how, things that could affect the speed, and what's going to be the end result. So now we could talk about some general things about neurons to kind of finish up now. For example, what if you get multiple signals? So one neuron, for example, could receive multiple signals from multiple neurons. Remember, neurons could communicate with each other in multiple places on that cell. So one neuron could potentially get a signal from multiple neurons that might be conflicting signals. So what do you do? What does a neuron do if it gets multiple mixed signals? Well, it has options like always. Something it's going to do to help come up with its own plan or analysis of information is called summation. This is basically how your neuron is going to listen to multiple signals at once to come up with a response. And like always, there are options. When it comes to summation, there are 2 major types. There's spatial summation, and there's temporal. What's the difference? Again, they're both how a neuron is going to listen to multiple signals. But how? What's the difference? They're both called summation, meaning they're going to add some signals up. And the major difference is how you're going to add up the signals. How are you going to tally the votes, basically? In spatial summation, it kind of gives you a hint. It's called spatial for areas. This is where a neuron listens to all the signals it's going to get in one particular area. That's what you're seeing on this slide. This neuron in pink, labeled number 3, is getting a signal from 2 other neurons. And it's only listening to all the signals it's getting, basically at its dendrites. It's looking at all the signals it's going to get within a period of time in one particular area. That is spatial summation. It's adding all the signals from one area. That is spatial summation. And so every time it gets an excitatory signal in a particular area, it's going to generate the excitatory postsynaptic potential, depolarizing, aka becoming slightly positive, until it hits threshold and it will make its action potential to send the signal. But again, there are other ways to do it. Your body also has temporal summation. Again, just break the name down. You're still doing summation. You're still listening to multiple signals. But how now? It's called temporal because you're now listening to all the signals you get within a period of time. So this neuron can now listen to all the signals it's going to get anywhere, but only within a certain amount of time. That is temporal summation. And again, every time it gets an excitatory signal, it will generate an excitatory postsynaptic potential, depolarizing, aka getting slightly positive until it hits threshold, and will finish off the whole thing, making its signal, making the action potential. So that's summation. So it doesn't matter if your neuron gets mixed signals. It could get excitatory and inhibitory signals. It'll just add them all up, and whoever has the most votes wins. That's all it is. And it's just whether or not you're doing it within a particular area, spatial summation, or within a particular time frame, temporal summation. And that's how your neuron will listen to multiple signals. And again, there are multiple neurotransmitters. There are multiple chemical messengers. Only one I want you to know is acetylcholine, the first one on this list. It's actually an excitatory neurotransmitter. So whenever a cell gets a signal from acetylcholine, it knows it's being told to do something. It's going to generate one of those excitatory postsynaptic potentials, depolarize, and if it keeps getting in enough signals, it will hit threshold and make an action potential and send a signal. And that's all I want you to know. There are other neurotransmitters. I can mention other ones, like on this slide. Some neurotransmitters send specific signals. Like this next one here called substance P. Substance P helps send pain signals. Kind of think the P in substance P is for pain. So again, when you're walking around barefoot and you stub your pinky toe on the table and life feels like it's over, you're feeling all that pain because your neurotransmitters are releasing substance P to let you know you're in pain. But again, stick to the exam review. There are some other more in-depth topics in here. Well, another one is called neural circuits. A neural circuit is just a group of neurons that are kind of gathering and providing specific types of information, but you do not need to know your different types of neural circuits, so you can skip over these. You do not need to know the different types. These are just pictures. A circuit literally is just a circuit, a group of these neurons working together. You do not need to know them. We're going to skip over them and finish off with this last topic for today. You basically know all about the microscopic parts of your nervous system, except for one last thing, is what happens if you damage it. You know about tissues. You know when you damage tissues, you could either fibrose or regenerate. And when it comes to nervous tissue, they're likely to fibrose. Why? You're not likely to regenerate it. How do you know? It's simple. If you were to sever the spinal cord, you'll become paralyzed permanently. You won't become paralyzed for a week or a month and then later walk. No. Why? Because you don't regenerate the spinal cord. Why? Because you don't regenerate nervous tissue. It has a limited ability to regenerate. Meaning basically, it's not. Why? It's because neurons don't really regenerate. And I say don't really because you can generate a neuron in specific situations. I want you to know the one situation where you might be able to regenerate a neuron. Well, there's one particular instance when you might be able to do that. It's if the cell body remains intact. That's what you see on this slide. You will only repair neurons, possibly, only if the cell body remains intact. Well, that basically means you could damage things like the dendrites and the soma. But if you damage the cell body, the neuron won't regenerate. When you cut through someone's spinal cord, you damage cell bodies. They don't regenerate. So when we talk about fixing or regenerating nervous tissue or neurons, we're really, in this case, talking about how do you fix an axon. Because remember, the cell body has to stay in place. And like usual, I want you to know the steps in the repair of an axon or the regeneration of a neuron. There are 4 steps. I want you to know them in order and what happens in each step. And you can see them all on this image here, this last image. Like usual, I'll name the 4 basic steps in the regeneration. And then I'll go back over and talk about what happens in each step. So let's imagine you are Again, maybe cutting up some vegetables for dinner and you slice through your finger, maybe you nick a nerve. Over time, it can possibly heal as long as the cell body's intact. And your body's going to go through 4 major steps to do it. In order, first step is called Wallerian degeneration and chromatolysis. The next step is called axon growth. Third step is called regeneration tube. And then the fourth step is the axon grows and reattaches to its target. So let's go through these 4 steps. So what's going on? There's something called, first step is Wallerian degeneration and chromatolysis. What's going on here? It's going to seem sort of counterintuitive, but this is what happens in your body. So again, we're just saying we have cut through an axon. How do we fix it? Well, when you cut through the axon, the first thing is going to happen is the distal segment of the axon will actually get broken down. Your body will get rid of it, meaning the part of the axon that is not attached to the cell body gets broken down. That's the distal end. While the part of the axon that is still attached to the soma, to the cell body, will still be there. When you break down this distal part of the axon, we call that chromatolysis and Wallerian degeneration. You're degenerating that distal part of the axon. Why? Your body doesn't use it. Your body will regrow the axon using part of the axon that was still attached to the soma. So that's step 1. Wallerian degeneration and chromatolysis is simply when you degenerate, when you break down the part of the axon that was cut away from the cell body. Then the second step is simple. The axon will begin to grow. That is the second step. The axon grows. But you can imagine the axon doesn't know where to grow towards because it was cut away from wherever it was reaching out to. So it needs some help to get to its original location. And it's going to get help from Schwann cells. Schwann cells are going to come in and make a little tube made out of myelin. And the axon is going to use this tube to grow in. And we name this tube. It's called the regeneration tube. That's step 3. So in step 3, you have regeneration tube formation. What happens here? Schwann cells make myelin to form a tube to guide the axon. And then once it makes the tube, you'll do your last step, which is the axon will finish growing and reconnect to its target cell. That's it. And like always, keep it simple. First step, Wallerian degeneration and chromatolysis. Break down the part of the axon not attached to the cell body. Step 2, axon grows. Why? Axon is growing. Step 3, regeneration tube formation. What happens here? Schwann cells make myelin to form a regeneration tube to guide the axon towards its location. And then step 4, the axon will finish growing and attach to its target cell. The axon will finish growing and go back to its original location. And that's how you regenerate at least a part of a neuron. At least the axon. That's chapter 12.